Good evening, everybody. I welcome you all back. And today we will do the last part of algae, in which I do the family of Rhodophyceae and Cyanophyceae. So I hope you are revising the bryophytes and the algae, the part which we have done earlier. So I think please work hard on these things because they will help you to complete all the studies part and clear the competitive exam. And this clearing the competitive exam is the best return gift you can give to me, right? So just be indoor, be safe. So I will just catch up you in few minutes for the last part of the algae. So stay on. Okay, welcome back. And now I will start the concluding part of algae. And this part consists of two families which are very important families, that is Cyanophyceae and Rhodophyceae. So before that, I will revise you what we have did. So we did the Chlorophyceae, and in Chlorophyceae I just told you the pyranite bodies are there in Chlamydomonas, and these parent bodies, they are surrounded by a sheath. And if sheath is not complete, it is incomplete, it is called polar pyrenoids, right? Then I just discuss you with the types of the algae, that is the banthophytes. Then we have the other type, epiphyllophytes, right? So today I will start the Cyanophyceae. The member of Cyanophyceae, which is also called Mixophyceae, are commonly known as blue-green algae, and as principal pigment is bluish green, that is phycocyanin. So the first genera in it we are doing is the Oscillatoria. Oscillatoria is basically unbranched filamentous alga with uniseriate filaments, right, with thickened membrane calyptra. And it is the example is Oscillatoria proboscidea. Proboscidea is the example. The main photosynthetic pigment is chlorophyll A, right? So and the accessory pigments, as you know, because of the color, it is phycocyanin and phycoerythrin, right? Which are present on the surface of thylakoids, as membrane bound plastids are absent. It's very important, right? In floating species, there is a tubular gas vesicles are formed in the cytoplasm. The gas vesicles are usually formed in low light intensity when filaments are near the pond. So you should keep in mind gas vesicles in the floating and when they are found. So we are coming to the next important part is the reproduction. So during vegetative growth of trichomes, some cells are filled with mucilage, right? And they become dead and called necridia and function as the separation discs. The cells of trichome between the necridia are called harmogonium. So the first type of reproduction is harmogonium, and to understand it, I just want to show the diagram. See, this is the diagram, and this is unbranched filament, and here some cells, they are filled with mucilage. They are called necridia, and they ultimately behave as separation disc, and in between them the cells they start behaving as a harmogonia. Okay, harmogonium. So this is the structure we are doing, and I also told you covering like that of calyptra, right? So I'm coming back to again to the name of the Oscillatoria, and the name of Oscillatoria is due to the oscillatory movements. So there are gliding movement, oscillatory movements, creeping movements observed in Oscillatoria. The trichomes of Oscillatoria move left and right, right, like a pendulum. See, right, like a pendulum, it is moving right and left, and hence, like a wall clock I showed you, hence called the name Oscillatoria, right? The reserve food material as a special type of starch, which is called Cyanophycean starch. Then chlorophyll A, when traps solar energy, and it changes into starch and glycogen, and which subsequently changes into the Cyanophycean starch and glycoprotein, right? So this is the oscillatoria. I just show you the oscillatory movements. See, pendulum-like movements, and because of these movements, they're called gliding or oscillatory movements. A1 is gliding and B is the oscillatory. And then we are coming to the next part, that is nostoc. See, in the nostoc, it is same trichome, and here in trichome there are present these heterocysts, and heterocysts are two types. One are the terminal and some are between the intercalary, in between the cells, and these which are intercalary, they have these two polar nodules, and they have the synophysin granules, and in between there are the cells which behave and do the asexual reproduction. They are called the actinids. And now we are coming again to the nostoc. Nostoc is a freshwater as well as terrestrial habitats. Nostoc grows in the rice fields, and the trichomes of nostoc, which I already told you, are the uniseriate, right? Characterized by presence of heterocysts, which are two, I told you, terminal as well as intercalary, and gives a beaded appearance, that is molliform appearance. It lacks nuclear membrane and nucleolus. This type is called, very important, I put a star, incipient nucleus. So question arises, where the incipient nucleus you will form, then you will say it is myxophysi, synophysi or nostoc, any option it will comes. The prominent is chlorophyll A, C phycocyanin, C phycoerythrin. So then chromoplasm contains 70S ribosomes, which shows that they are the prokaryotes. I will take later on difference between the rhodophysi and synophysi. Nostoc punctiforme is found in the corallite and anthoceros. I have just told you earlier also in last lecture, endophytic fungus, and these endophytic fungus are given a lot of examples. So, but Nostoc punctiforme is one. So intercalary in heterocyst, just I show you, has two polar nodules, and terminal one has one, which I explained you. Now the reproduction part, it is same that of oscillatoria. Harmogonia, the filaments break at different points, and each broken filament due to heterocyst is called harmogonia, right? And by actinate, I just show before starting this, during unfavorable conditions, some cells of filament become enlarged and covered by thick wall, and it is called actinates. We also call arthrospores and resting spores. Heterocyst, I explained you, terminal and intercalary, and depending upon that, the intercalary, which are present intercalary, and they have the two polar nodules, and the terminal one has one. And heterocysts are also acting as a resting spores and behave as a normal reproductive cells to form a new filament. And endospore is the next, another type of reproduction, and the two sexual reproduction and sex organs are absent. And this is very important. When I do the difference between the rhodophysi and synophysi, I will explain you these all things, right? So now we are coming to the next family, that is the rhodophysi, right? The member of class rhodophysi are commonly known as red alga due to presence of, please mind it, water soluble. Question will come whether it is water soluble or non. Phycoerythrin. Other pigments are chlorophyll A and D. This is very prominent. So see rhodophysi. Rhodophysi, we have D, and from here it shows that rhodophysi has D, and the chlorophyll D is present. Besides, the chief xanthophyll is teraxanthin, and also new xanthin is present. The thallus organization is of two types. That is uniaxial thallus, if the branches from a single filament are involved, or is called uniaxial, which we are doing this time, batrachospermum. And the cell of thallus are uninucleate with discoid chromatophores, right? And multiaxial, which we also call the fountain type. Here, if the branches of many axial filaments aggregate together, and the example is polysiphonia. But both are called pseudo parenchymatous habit. Pseudo means false parenchymatous. So here it is very important to tell you there are two forms. One is primitive, which is subclass Bengoidae, have a stellate chromatophores with paranoid. But in Bengoidae also the paranoids are naked. Whereas in advanced form like Floridae, which consists of batrachospermum the discoid without any pyronates. This is very important, right? So we are coming to the batrachospermum. Batrachospermum consists basically of fresh water. 98% of rhodophysi are marine, but only the batrachospermum, which we have studied from long ago, so it is the fresh water, right? The thallus is uniaxial. I already told you uniaxial. So batrachospermum molliforme found attached to the stones on the swift flowing fresh water, right? So mature thallus is branched and differentiated into the prostrate and erect system, right? So the main axis is differentiated into the nodes and internodes, and from nodes develops two types of branches. The branches of unlimited growth show monopodial or pseudo-dichotomous type of branching, and the branches of limited growth results in a globose mass called glomerule, right? So I will explain you like this here. See, this is basically giving the prostrate and erect. Prostrate is help in the anchoring, whereas erect is further divided. When we will see erect, so you will see here it is the laterals of limited growth, and these are the long branch of unlimited growth with apical cell upward, right? So this having a when we see, so these limited one, they will together form a structure called glomerule, right? This. So then we again come back to our branch, that is the reproduction part. So in reproduction is the asexual reproduction, which is by means of non-motile spores, and that is monospores. Monospores, which are the spores which develop singly in the monosporangium. And the second are the carpospores, very important. So here the diploid zygote undergo meiosis, and it will form haploid nuclei. And these haploid nuclei, here they formed, and then they are transferred into these gonomoblast initials. And then gonomoblast initials ultimately results in the formation of gonomoblast filaments. And on the tip of the gonomoblast filaments, like this, are formed the carposporangium. And in carposporangium, because meiosis will occur, in carposporangium, the forms are the haploid carpospores, right? So then we are coming to the next part, that is sexual reproduction. So in sexual reproduction, the flagellated gametes are absent. If you remember, in my first lecture, I told you that in advanced oogamous type, only in case of rhodophysi, the flagellated gametes are absent. So it is here we have the spermatangia, which is male reproductive organs, and it is unicellular and uninucleate, spherical or globose, colorless structure, produced singly at the distal end of the branches of limited growth. These are those branches which together form a globose mass, which is called glomerule. I just told you, right? So and then we have the next is the carpogonia, which is also called procarp, female reproductive structure, flask-shaped structure differentiated into the basal swollen egg, like this, right? And long, narrow, this is called trichogyne, right? It is also like spermatogonia is uninucleate, and trichogonia receives the male gamete, because male gamete is not motile, so it receives through the water currents. So male gametes, spermatogonia, and female egg, they fuse, right? They fuse to form a zygote, same as what did here, same. Zygote is formed, and the further process is same. Meiosis occur and haploid nuclei form, 1, 2, 3, 4. And these haploid nuclei, they are shifted to, I already told you, these haploid nuclei, they are shifted to gonomoblast initials. And gonomoblast initials, we have the gonomoblast filaments. And on the tip of the gonomoblast filaments, we have the carposporangium, and in carposporangium are present carpospores. And these lowermost cells of gonimeblast filaments, hey, this one, they will form this one, lowermost cells, they will form a structure called lowermost cells, they will form structure called sterile filaments. So these complete structure, which consists of sterile filaments, gonimeblast filaments, carposporangium, in carpospore, a complete structure of carpospore, gonimeblast filaments, sterile threads, they form a structure, very important structure called cystocarp. See, this structure is formed, and you will see, and here the carpospores, these are the sterile threads and gonimeblast filaments, they together form a structure called cystocarp, right? So this is a very important structure, and here it is very important to mention that on germination the carpospore form a branch heterotrichous filamentous structure called Cantentia stage. This stage is very important, and you will see Cantentia stage, right? So this is an important stage we are doing, and this is Cantentia stage I am telling you, and this Cantentia stage produce monospores, which are the means of asexual reproduction, which I have already told. And so this life cycle of batrachospermum, where the two haploid phase, one is the gametophyte and the other is the carposporophyte, they alternate, and with the diploid phase, right? And that is diploid phase is zygote. Such a life cycle with two haploid phase is called diphasic or haplophasic life cycle. So this I finishes the rhodophyceae, which I just want to explain. So now I am coming to the last part, that is the difference between the rhodophyceae and cyanophyceae. First, as I already told, rhodophyceae is eukaryote, cyanophyceae is prokaryote. And in rhodophyceae, as D is there, so we have chlorophyll D, new guest beside chlorophyll is present in both. Whereas we have heterocyst absent in case of rhodophyceae, but it is present in case of cyanophyceae, because we told you that we have seen in Oscillatoria and nostoc we have seen it, and Golgi bodies, and we have the endoplasmic reticulum are present, whereas they are absent in case of the cyanophyceae. And the true sexuality, I already told you, present in most members, but it is absent, whereas that role of sexual reproduction absent, I told you earlier when I doing there, see, sexual reproduction and sex organs are absent in nostoc. So it's taken by the parasexuality. So then we have to do chromatophores with single thylakoids, and I already told you these pyrenoids are basically naked pyrenoid present in Bengoidae, but there are no pyrenoids in case of subclass Floridae, which consist of batrachosperm, right? But pyrenoids are absent in case of cyanophyceae. And then last, which we again said, that chlorophyll D is present in rhodophyceae, whereas absent in the cyanophyceae. So this I finishes the last part, and I also again tell you the pyrenoid, which are characteristic feature of chlorophyceae, and these pyrenoid, they are surrounded by the starch sheath. And so I finishes, and I will back with the other part subsequently. So please revise it and try to work hard so that you can clear the competitive. And please remember that clearing that competitive exam is the best gift you can give to me as on the screen. Thank you. Bye bye.